So hello, welcome to this video from Mr. Gill's Science Stuff. This is the second video of 3 on the coronavirus. This one we're going to take the difficulty up a little bit to about good GCSE level. So if you haven't watched the Key Stage 3 video, do watch that because we're going to address different questions in this one. And some of the questions you might have been answered in that first video, things around naming and so on and so forth. So please do watch that one before you crack on to this. So in this video, we're going to answer 10 questions. Okay, there they are. I'm going to try and rattle through these reasonably quickly, but compared to the last video, there's a little bit more density, a little bit more depth, a little bit more data, a little bit more difficulty than there was in the first video of these 3. So do feel free to pause, go and look at the sources. All the sources are at the end of the presentation and in the description. So, alright, cracking straight on then with question number 1. What is so special about this disease? Now, obviously, we're trying to address the biology of coronavirus in this rather than a lot of the media hype. And the answer is really coronaviruses. There's not a lot that's actually that special about them. They're one of a fairly normal family of viruses that circulate and cause upper respiratory tract infections, along with picornaviruses, rhinoviruses, adenoviruses, that kind of thing. The thing that's new about this one, and we covered this more in the first video, is this one has got new antigens. Now, I am assuming that you understand what we mean by that term. So this one's got new antigens. It's a new variant on the outside of its the viral particle there, which means we have no immune to it. And as an interesting point, coronaviruses don't use DNA in their genome. They use RNA instead. They are RNA viruses. So that's question number 1. Number 2, how do I become immune and how do I recover? Well, we covered a lot of this in the Key Stage 3 video, so do go and recap that. I'm making the assumptions in this video that you know the following terms: phagocyte, lymphocyte, antigen, and antibody. Okay, so as a very fast recap, in the previous video we looked at how the immune system works. So here's our virus. Our virus gets picked up by the immune system, particularly a type of cell called a lymphocyte, so a white blood cell called a lymphocyte, which then stimulates the activation of other lymphocytes that create antibodies. Those antibodies get created, they get released, they are specifically targeted at this virus. When the virus is within the body, they stick to the virus, to the antigens on the outside, they glue the virus together, stopping it infecting your cells and holding them all together so that another form of white blood cell, the phagocyte, can come along and consume the lot. Okay, so that's a very quick recap. So where do vaccines come in? What is a vaccine? Well, essentially, how do we make people have that response, have the ability to make the right antigens for the long term, or the right antibodies for the long term, without actually making them sick? Okay, well, this was the first 2 steps of how the immune system works. Now, really, it's only the antigens that the immune system is actually working against. So what if we were able to take away the virus and just put the antigens in? Okay, well, then our lymphocyte would force the generation of antibody-creating lymphocytes that would form these antibodies, okay, so that much later, right, when the real virus came along, there it is, these antibodies are produced very, very quickly. Okay, those antibody-creating lymphocytes that stick around after a vaccination or an infection are called memory cells, and they provide you with what we call immunity. So that's the aim. So essentially, we skip straight from infection to the agglutination bit, the bit where we're sticking the viruses together and the phagocytes coming along and consuming them very, very quickly, so they can't infect many cells and they can't make you sick. And that's how a vaccine works, right? So you think, okay, great. Well, why can't we just bust up a few coronavirus particles and stick them in someone as a vaccine? Well, there's actually no guarantee that just the antigens is going to, give enough of an immune response to generate enough memory cells to provide immunity. So what we need to do is we need to trick the immune system into believing that you are actually infected, right, while not actually making you sick. And there's lots and lots and lots of different ways to do this. And I'll go through a couple of examples, and there are many being tested for COVID at the moment. So if we don't have that particular response there, we don't have the antigen providing enough response, we don't get the production of memory cells, and we don't get our wonderful immunity, which is what we want. So Can we make a vaccine? And the answer is yes, definitely. Many groups are working very quickly on this. This is the fastest generation of vaccine candidates that I think the world has ever seen, because we know how to make vaccines. We do it for seasonal flu every year. Vaccine production is quite a normal pharmaceutical thing, but it still needs to go through rigorous testing so that it does a number of important things. So when we think about it, what should a vaccine do, a good vaccine? Well, it should provoke the immune system enough for long-term immunity. So when you really get infected with anything, it causes all sorts of damage and inflammation and swelling and things like that, which alert the immune system and generate a very strong immune response. Now, we obviously don't want our vaccines damaging you, but we do want the immune response. So we want to provoke the immune system enough for long-term immunity, but we don't want our vaccine to be infectious or pathogenic in its own right. So it's a balancing act to strike there. Okay, we don't want it to harm the patient. We don't want it to we want to minimize side effects. Now, you might know after some vaccines you might have a very small amount of fever. That's your body's response to infection. You might feel a little nauseous. Now, with each vaccine you have to weigh up what is better, what is worse, the very minimal side effects that you get versus the long-term immunity to a deadly disease. Okay, and obviously it's much better to be immune to a deadly disease and just take any side effects on the chin, as it were. But we do need to minimize them in the development stage. Also, we need our vaccine to be stable, transportable and long-lasting, because once we get this vaccine sorted, it's no good if you can't transport it out of the laboratory fridge and get it to the front line where it's needed. Okay, so that's what our good vaccine should do, which is why it is not a quick process to just go, yep, we know what virus is causing this, here's the antigens, off we go. So you may see seen graphs like this. This is the first of many graphs in this talk that's going to be. So in the first instance there, the so-called primary vaccination, we stick our vaccine in there, and yeah, it generates some memory cells, some antibodies. But let's say we need about this level here for for long-term sustained immunity, and our initial vaccination doesn't produce that. Well, that's why some vaccines have booster injections. Some vaccines you need 2, maybe 3 doses of the vaccine to get enough immunity to be long lasting. Okay, once you've got that, then you get a much stronger response and our memory cell levels stay nice and high. They do decline over time, which is why some need long-term boosters. Think tetanus. If you injure yourself in the garden with all the spare gardening time that you've got this Easter holidays, then you will be asked, if you go to the hospital with a nail through your hand, when was your last tetanus injection, because the memory cells for your tetanus vaccination will be dropping over time. Okay, so moving on then. So specifically for COVID, there are 2 vaccine candidates that are in phase 1 trials. By phase 1 I mean actually being put into people, and 42 vaccine candidates in preclinical trials. And these are people doing different things with different bits of the virus, inserting them into different what we call vectors, things that transport those antigens into somebody's body. So that is the virus itself, the structure of Covid-19 or SARS-CoV-2. As you can see, it's got those spike glycoproteins, and those spike glycoproteins we know from other coronaviruses produce a very strong immune response. So it was actually the 17th of March that it was reported that the first US volunteer tested the first vaccine. And this was a very simple vaccine. You can obviously read that, pause now and read that if you want, but the link is at the end and in the description. This was a woman from Seattle, a 43-year-old mother of 2 by the name of Jennifer Haller, who actually got vaccinated with a copy of the genetic code of the virus. So they literally put a copy of the RNA to see if that would work into it. Now that's a very quick process, but will it generate enough immunity to be a useful vaccine is the question. And the answer is we don't know yet. Okay, it's still in the testing phases. So another example I'll give you, and there are loads of examples. The link to all of the current examples is in the description. But this is one example that jumped out to me from the Oxford Vaccine Group at the University of Oxford, which is to use modified chimp adenovirus. So chimps can suffer with colds and coughs as well as we can. Adenoviruses are just a family of viruses that cause those things. So this virus is called ChAdOx1, chimp adenovirus Oxford 1, and this has been used as a vector. So what we mean by a vector is the group there has managed to get this chimp virus, which can't infect us, doesn't replicate in humans, to carry those spike proteins into a human. Now this is shown to be effective in a dish, right? but that's always a very, very tentative, hopeful sign. If you see a science paper that says, this thing kills viruses in a dish, bear in mind, so does fire. Doesn't mean you want it in your body, right? So this is it. It's one of the preclinical ones, but it is hopeful because chimp adenovirus has been used as a vector to carry other antigens into humans and been successful making other vaccines. So onwards, number 4. What about other treatments then? So can we just treat the virus as and when people get affected? Well, yes, there are other treatments being looked into. Plasma transfusions are quite a direct one. So if you are a healthy person who has recovered from COVID-19 and you donate blood, well, usually what gets taken is the red blood cells when you donate blood. But within the plasma, because you've already made those memory cells, those lymphocytes producing those antibodies, will be a load of antibodies for COVID-19. So the idea is maybe we could just use those as a temporary stopgap in people who are really, really suffering, whose immune systems aren't quite catching up as fast as the virus is replicating. Obviously, there's issues with that. If you put foreign proteins into a patient, then there's all sorts of other issues as well. What if their immune system reacts to the treatment rather than reacting to the virus, which is a problem. But that's one idea. There is a way of mass manufacturing antibodies to specific things, so called monoclonal antibodies, and we'll get to that in a little while. There are also lots of existing drugs out there on the market which may have some activity against SARS-CoV-2. Great advantage of using existing drugs is we know they're safe to put in the human body. So that really, really abbreviates the clinical trial stages. It really, really abbreviates how much trial you need to have. Okay, so you've got to be careful with these though, right? Because people with, shall we say, limited medical experience, like the dear President of the USA, starts touting various drugs and saying, yeah, this is brilliant, because he's not really abreast of the scientific information, if we're honest. So this one, hydroxychloroquine, which is touted as a cure for COVID-19, he doesn't know that. It's being tested, and actually a lot of the evidence suggests it's really not actually that useful at reducing the duration that people are infected with this virus. So one page you can look at, again, link in the description, is the European Medicine Agency, which is listing all of the different medicines that are currently being investigated for activity against this particular virus. And there we've got a variety. There's 5 there. So these are the potential treatments, and they're undergoing clinical trials to assess their safety and efficacy against the disease. Okay. Now, obviously, you can go and look into what they are. We've already mentioned the anti-malarial treatments there that Trump was touting around. There's anti-HIV medicines. The logic there is that HIV is also a virus that uses RNA, so maybe there's some similarities there. That's being investigated. Then, of course, we've got things like monoclonal antibodies, which we'll get to in a moment. Okay, so moving on. So if we can't reliably vaccinate yet and we can't reliably treat yet, surely it would be a good idea to test for the virus. And if so, how? Can we test for the virus? And if so, how? Yes, we can. There's 2 ways. We can look for its genome. Right, we can look for if its genetic information is within your body. So take a nasal or throat swab. Tells you if you have the virus in the system. More usefully, we can look for whether or not the person has actually made antibodies in response to it, which tells you if you're immune. Okay, and that obviously is very useful because, as we said in the previous video, if you are immune to this allows you to be more active in the community. You can come into contact with people who have the disease, and you can end up being more useful, being more helpful, and particularly healthcare workers who are isolating can go back to work, right, which obviously is very important. Now, this is one of the hardest parts of the GCSE syllabus, right, which is the formation and use of monoclonal antibodies, but it is right at the heart of current debates about COVID. So monoclonal antibody means one type, okay? And this diagram over here, as you can see, it shows how we produce monoclonal antibodies. So what we have to do first is we have to find the protein that we want to make an antibody against, and that's been done. Right, so COVID-19 has been characterized. We know what its viral proteins look like. We know what its genome looks like, so we can get its proteins no problem. And we need to inject those proteins into something with a lovely immune system that will be helpful to us. So in this case, usually it's a standard lab animal, a mouse or a rabbit. You could use a leopard or a puma, but the risks to your personal safety would be quite high, and I think the animal welfare people would have even more to say about it than they do already. So these are lab animals that are bred for the process, and their immune system obligingly generates white blood cells, generates these immune cells that produce the antibodies against the protein that you've put in. So you've now got antibodies against whatever antigen it is. So over here, look, what we have to do then, unfortunately, is get the antibody-forming cells from the spleen of the animal, which does unfortunately mean the end of that animal's existence, and the bits go in a blender, and then we get the bits out that we need. Now, the thing about animal cells, though, is they don't grow very well outside of the animal's body. So what we need to do is fuse them with a cell that does grow very well outside of the body, and that's what we would call a tumour cell. Now, there are various lines of tumour cells which are fascinating as to where they came from. I recommend going and look into HeLa cells, H-E-L-A. Henrietta Lacks famously was the donor there, and some questionable medical ethics stuff. So if you're interested in that, go and look. There's a book, there's a film. So we take some tumour cells grown in a lab, we take this spleen cell from the animal that's making the antibodies that we want, and we fuse them together. And hopefully what we end up with is we end up with a cell that can both grow outside of the body, like a tumour cell can, and it can produce the antibodies like the spleen cell, like the lymphocyte can. And this is called a hybridoma. So the mouse makes the lymphocytes, tumour cells can grow outside the body, we fuse them to make hybridomas, and those hybridomas growing on a dish are basically little antibody factories. So you grow them on a dish, and then you can harvest the monoclonal antibodies. And monoclonal means you've got antibodies specifically for one thing. Okay, you've got them against one thing. In this case, it would be the spike proteins, probably, that stick out of Covid-19. So, use of the MCABs, the monoclonal antibodies. So generally, we could treat directly. We could put these into a person and treat the disease itself, or more likely, and what's been talked about more, is testing. So antibody testing is actually really common. You can buy antibody-based tests from the local pharmacy already. They're really normal. The most famous one, of course, is the pregnancy test. Pregnancy tests work on monoclonal antibodies. Okay, that's a diagram for how a pregnancy test works. As you can see, in a pregnancy test, we're not looking for proteins from a virus, we're looking for a hormone that's only produced during pregnancy called HCG. You've got a monoclonal antibody here with a dye attached to it. If that monoclonal antibody attaches to the molecule you're looking for, right, and moves up the stick like this, then it will bind to a second type of monoclonal antibody here in this little window and produce that famous blue line, which indicates that the pregnancy test has worked and is positive. In this instance, it wouldn't be a pregnancy test, it would be a test for SARS-CoV-2, the proteins thereon, and then you've got this final window down here which has a monoclonal antibody that attaches to the first monoclonal antibody, which is a control window. All that does is just prove that your liquid made it from A down to D. Okay, so the World Health Organization is very, very keen on this, and countries that have been successful in containing coronavirus, containing COVID-19, have been doing very, very extensive testing. So think South Korea, increasingly think Germany. They've been doing very, very, very good work in terms of testing. The message there being test, test, test from the World Health Organization. In the UK, we've not been so good at this. I don't really understand why. Our government hasn't done really what it's supposed to do in terms of obeying this. It would be very, very useful, particularly for letting people go back to work where they work in key frontline roles. This was a news story that came out yesterday in the Times. Again, link is in the description. That a few weeks after we should have been getting onto this, when we finally got onto it, Britain has bought millions of coronavirus antibody tests, but they don't actually work. Right, they're not actually specific enough or reliable enough to tell if somebody has got coronavirus. Because there's one thing worse than no test, and that's a test that tells you you are negative when in fact you're positive. So really can't risk that. So again, have a look at that if you can. So we've obviously all sat at home, which is why we're all doing these videos, why I'm sat here talking to you from my house rather than doing this in school. Herd immunity versus flattening the curve, right? You might have heard both of those terms. What do they mean? And I would highly recommend you go and have a look at the Washington Post. There's a famous article they've put out for free now, which basically models what happens if a population is contained. So this term herd immunity is an important term. It means enough people in a population have had the disease that the rest of the people, well, they're not going to pick it up from anyone because the majority of the population are immune. Famously, this man here, Dominic Cummings, who is advisor to Boris Johnson and by all accounts a, should we say, morally questionable individual, argued that really we should just let coronavirus go through the population. And there are some quotes taken here which were reported first in the Times, and you can go and look at the articles yourself. He's quoted as saying, "If that means some pensioners die, too bad." And there's some other quotes. So that is one method, right? Is just to allow a disease to move through a population. And sure, herd immunity will build, but the numbers of people that would get ill and die at the same time would entirely overwhelm our system, which is where this term flattening the curve comes in. And this here is a wonderful set of animations about what happens if we just let diseases run rampant through the population versus actually do something to try and control them. So all the little dots here are individuals, and as you can see, all the people who are brown are people who are infected with the disease. Now this is a fully mobile population, moving around, mingling, going to work, going to school, so on and so forth. After an amount of time, the people in purple are the ones who are immune. So as you can see, we end up very, very quickly getting a large infected population, and then we end up with a very large infected population, and then eventually some immunity. But that spike is very, very big. If, however, we do some quarantining, we do some social distancing, right, where we keep people maybe in one country, stop them moving, right? But obviously, if those people are able to move in and out, so this is, for example, China, then you can see where we get different spikes of infection at different times in different countries. Okay. However, if we close the borders, then that would not have spread so fast and so far. Okay. So social distancing is the next one down here. So this is the example if we look at 3/4. So this is a quarter of our population continues to move around, while the other 3/4 adopt a strategy of what's called social distancing. So the blue dots that are still are the ones who are sat at home social distancing. As you can see, that brown amount of people who are ill rises still quite quickly, but not as fast. And the immunity comes in, and then there's an amount of people who haven't had the disease. That is far more manageable for any health system. Okay, if we move further on, this is where we're looking at 1 in every 8 people is allowed to move, okay, and the rest of people stay at home, okay, which, again, I'm not sure exactly where the figures are for proportions of people who are allowed to move in the UK. That's another debate that we can have with the politicians. So, as you can see, look, the disease doesn't spread anywhere near as quickly. The amount of people who are sick is quite easily manageable by the health service, okay. So the different simulations that we've just run there, and this website does run a new simulation every time, shows the difference between a free-for-all, attempted quarantine, so that's basically trying to keep it in one country, moderate distancing, and extensive distancing. And we're kind of somewhere in between these two on the right here, okay? Now, what we're really trying to do is keep the peak of this curve below what the National Health Service can deal with. Okay. Now, and in the meantime, also increasing what the National Health Service can cope with. So this is our flattening the curve. The dotted line there is healthcare system capacity, and if that line is exceeded, it doesn't mean that just people with Covid-19 aren't getting treated. It means that people with other health conditions aren't getting treated either, and that's obviously a very bad thing. So that's what we mean by flattening the curve, and that's what we're trying to do. Now, this is our part of the bargain, staying at home and staying safe. The government and the healthcare system's part of the bargain is to make sure that they're doing everything they can to raise that bar in terms of PPE, in terms of testing capacity, in terms of hospitals, and so on and so forth, which is why you're seeing the Nightingale hospitals being built, and hopefully at some point we'll get some mass testing. So, Social distancing obviously can be incredibly effective because this disease, for each person who is infected, on average, if they're just moving around, they will infect 2 others. So the amount will be doubling a lot, and we'll get to exponentials in a moment, doubling every generation. If, however, this person didn't meet up with this person, look at all these people who potentially could have been infected that aren't. Okay, so that's where we get to the numbers, we get to epidemiology. We're not going to go into this in a huge amount of detail, but I do highly recommend having a listen to the last few episodes of the BBC Radio 4 programme More or Less, which is a statistics programme and has looked in more detail at the numbers. I'm not going to go through everything they've said, but some of the questions that the things that have come up in the news that aren't necessarily clear unless you know what they're talking about are lag times, exponentials, demographics, and population densities. So we'll have a quick look at some of the graphs. So you might notice now that we've been in isolation for a number of weeks now, a couple of weeks, and the death count, as it were, keeps going up. Well, that's basically because of a lag in this disease. One of the dangerous things about Covid-19 is it has a long asymptomatic lag time, and there's lots of evidence there can be asymptomatic infectious people walking around the population. So you go from no symptoms, 5 days-ish, that may last up to 2 weeks, we don't really know, to fever and cough, to breathlessness, to ICU, to death or recovery, to reporting. Okay, so the lag time between any potential deaths and any potential infections can be up to a month, right, which is a long time. So if you think what was happening a month ago from now, people were still moving around quite freely, some sporting events were still on, think Cheltenham Gold Cup, so on and so forth, and our leader, Boris Johnson, we wish him a speedy recovery, but he was going to hospitals with Covid-19 victims in them and gloating about shaking everybody's hands. And now, of course, he's in intensive care. You can see how one thing led to the other. So don't get the impression that our social distancing strategy isn't working, right, because the death rates are still going up. Right? There is a massive lag time in here. So obviously death rates are still rising, on trend, doubling every few days. These are all graphs taken from the BBC, and these were taken yesterday, which was the 7th of April, but obviously it's a rapidly changing thing. So as you can see, look, there is an upward trend in death rates. We're seeing some slowing down of that. Again, we should see more of that coming through the longer we all stay in isolation. The more useful measure is to look at the growth in new cases, which actually is plateauing off. There are still new cases, but they're not going up with this kind of doubling effect, which shows that maybe tentatively the social distancing that we're doing is having an impact. Okay, and this is basically from the 1st of March to the 1st of April. Okay, now look at these axes here, they are log scales. Okay, so this is going up by factors of 10, not by factors of 1 or 2. Okay, so this is a log scale, so that's how this graph has ended up being flat, and hopefully you know enough maths to know what a log scale is. Okay, because if you don't make a log scale, this is what you get. You get these rapid increases, doubling, and then you can see the lag time between cases and deaths. Now, of course, we went into lockdown about there. Schools closed about there, and that's essentially only a couple of weeks before right now. So obviously a lot of these deaths here will have been infected before this lockdown started. So don't think it isn't working, right? We don't have the evidence for that yet. One interesting piece of data that has come out about probability of mortality with Covid-19 is that your percentage of dying from this disease, likely probability of dying, is the same as 1 year's total risk, which I found particularly interesting. And what that means is if you are a 15-year-old, for example, right, somewhere in there, 15 to 20-year-old, then your risk of dying in any 1 year from something, just over time, is between 0.01 and 0.05%. Right? That's roughly the same as our mortality with Covid-19. Okay, so as your age goes up, the number of the chance of your dying goes up, but it's the same as your standard risk of death, which I just thought was an interesting correlation. And obviously there's a generation of maths teachers out there who are absolutely looking with glee at these exponential graphs and going, well, our subject has come to its fruition. So when they were telling you, you will have to know what exponential graphs are in class, that was absolutely true. And if there's any small positives we can take out of this, it's a general population upskilling in terms of maths abilities would be a good thing. Right, Demographics and densities. There's a lot of comparison. This is going on in Italy, this is going on in Spain, this is going on in the USA, so on and so forth. Well, they're not actually direct comparators to us. They're useful, but we have to take those things with a pinch of salt. Italy's got a very elderly population. It's got the second oldest population in the world after Japan. USA has a very different political structure to us. They have state versus federal rules. Federal rules is the entire country. State rules, obviously, is within each state. So they operate quite independently and quite differently. And getting any sort of funding to do things on a whole country level is a bit harder. They also don't have nationalised health service, right, like we do. They therefore probably don't have centralised reporting, and getting the figures is higher. It was harder, rather. And, of course, they've got Trump, who is probably the worst person, the worst leader to be in charge of this that one could think of. China, of course, has very, very strong state control. They're on the other end of the spectrum, which means if they want to tell you you're staying in your house from now, tough, they can. So very, very strict government control of a lot of things, which might explain some of the successes in Wuhan and central China. They also have state reporting of disease cases, and they're saying there's no more diseases, no more new infections coming up from Covid-19. Whether that's true or not, we don't know, because the state does tend to massage figures. And obviously the UK is a very small country with a population concentrated in certain areas, the West Midlands and London and so on and so forth. Right? All these things make a massive difference. So yes, comparisons to other countries are useful, right? But they're not the be-all and end-all. They need to be taken with a little pinch of salt. So this leads me to question 8. Who do you trust about these diseases? Well, first off, check the source. Okay, if you see an article about COVID, about coronavirus, there's lots of them out there. Check who has written it. Ask what is their incentive for writing this thing. Right, following the money I think is quite useful. Who's paying them? Are they getting paid by ad revenue for clicks? In which case they're going to be much more likely to make it sound as dramatic and crazy as possible and act as unscrupulously as they possibly can. What's the motivation? Are they just trying to stir up controversy? Are they trying to push some particular ideology? Are they trying to educate you? Are they actually genuinely interested in public health? Right? Look for those sources. Okay, where have they got their information from? As there's been an awful lot of fake news going around on this. So trusted medical sources, ones I'd recommend would be the World Health Organization, European Medicines Agency, NICE, NHS, Centre for Disease Control and Prevention, and then our more reputable scientific journals like the Lancet, Nature, so on and so forth. Okay, those are the places to get the information. Now obviously there's a lot of obvious nonsense out there which you may have seen. For example, all this nonsense about 5G mutated a natural virus to make SARS-CoV-2. Obvious nonsense, right? Mobile phone antennas do not produce the type of radiation. They produce radio waves. They do not produce the type of radiation with anywhere near enough energy to mutate DNA. It is impossible. No debates, no conspiracy theories. That is not happening. Okay, none of this. Vaccines cause whatever, insert insane thing here. Sorry, anti-vaxxers, we have absolutely no time for your nonsense now. Right? There isn't opportunity for that. And obviously, people can have their own opinions and all this kind of thing. There is no time for that nonsense at this point. We have a global pandemic. Vaccines work. Get over it. Right? End of. This was made in a government lab is another conspiracy theory. First question, why? What would be the motivation? And also, if a government anywhere in the world could do something that elaborate, right, and that complicated and that diabolical, with nobody finding out, they're a lot more competent than most governments around the world seem to be. So this is obvious nonsense. A simple digging into the sources, checking the background of these claims, would obviously suggest that this is madness. Okay? We do not have time for it in the current state of affairs. We've got to look for proper information, as it's very, very important. And that's why we've got tech firms like Twitter, Facebook, Instagram, WhatsApp being summoned by the government to do more over crackpot 5G conspiracies particularly, and that's why WhatsApp has introduced this rule that multiply forwarded things through WhatsApp will be stopped. Okay? Which is a bit of a broad instrument to deal with that stuff, but fair enough. Number 9, when will this end? Well, we have no idea. Unfortunately, it depends on our adherence to the rules. So what needs to happen now is obviously testing, extensive testing like Germany. Personal protective equipment needs to be more readily available for the NHS, obviously. Contact tracing, which means following up on people who've got ill, who they might have been in contact with. Right, that requires people to do that job, very important. Isolation and treatment, and of course, more social distancing. Now, obviously, the more that we push our luck with the social distancing, the longer this will last. The more we obey the rules, the long it will last. Okay. Now, obviously, we have still got politics going on. We're on the recess at the moment. There's no government. They're on their Easter holidays. Boris Johnson's in hospital, which means Dominic Raab is in charge. We've got a new leader of the opposition who are having all sorts of debates. This was just one example. From a couple of days ago, the idea that Keir Starmer is saying that key workers are being overlooked and underpaid, which is probably true in a lot of cases. Our nurses, our healthcare professionals, all these kind of things are doing an incredible job at the moment, and we owe them as much support as we can give them, really. So, number 10, what should you be doing now? Okay, well, stay home, obviously. Keep fit. This is a respiratory tract infection. If you get it, a strong heart and lungs is going to make it much more bearable. Practice good hand hygiene, of course. Soap destroys virus particles, as we said in the last video. Be alert to the symptoms from trusted sources. Help those that you can help. So keep up with your work. Phone older friends and relatives. Loneliness from this social distancing can be a real problem. Mental health issues can be a real problem. So do make sure that you think compassionately and try and check in with people, and get tested if or when offered. So keep yourself busy, keep yourself creative, go and do something. And as I'm going to practice what I preach, here are all my sources for this video. Right, on that note, I'm off to build a greenhouse. Thanks very much.